The nervous system is a network of specialized cells that monitors the internal and external environment, processes information, and initiates commands through which the body reacts. Nervous tissue is composed of two types of cells: neurons, or nerve cells, and glial cells. A neuron, the basic functional unit of the nervous system, is specialized to conduct electrical impulses. Neurons have several important functional properties: a high metabolic rate, most are incapable of cell division, and they typically function throughout life. The size and shape of neurons is variable, but they share basic features. The cell body, or soma, receives, integrates, and sends nerve impulses. It typically contains a large nucleus that often has a prominent nucleolus. Ribosomes are abundant and form clusters known as chromatophilic substance, or Nissl bodies, that stain darkly with basic histological dyes. Two types of processes project from the cell body: axons and dendrites. Axons direct information away from the cell body. Most neurons have a single, long, thin axon connected to the cell body at a region known as the axon hillock. The axon generally does not branch, but it may have occasional side branches called collaterals. Most axons terminate as multiple fine branches called telodendria. These have distal expansions known as synaptic knobs, also called end bulbs or terminal boutons. Dendrites direct information toward the cell body. They are relatively short, tapered processes that branch along their length. They often possess small, irregular projections called dendritic spines, where axons can synapse. A synapse is where information is transferred between nerve cells, or to an effector cell, or to a sensory receptor cell. Glial cells, which are also known as neuroglia, are support cells that represent about 60% of cells in the nervous system. Four types of glial cells are found in the central nervous system, or CNS: astrocytes, ependymal cells, microglia, and oligodendrocytes. Two additional types of glial cells are found in the peripheral nervous system, or PNS: Schwann cells and satellite cells. Astrocytes are the most abundant type of glial cell in the CNS. They are star-shaped, with numerous branching processes whose expanded ends contact and support neurons. In some astrocytes, these processes contact capillaries as perivascular feet that help form a blood-brain barrier. This regulates the passage of substances between the CNS and bloodstream. Other important astrocyte functions include regulation of the chemical environment and promotion of neuronal development. Unlike neurons, astrocytes are capable of cell division and can replace dying neurons with a glial scar. Ependymal cells are epithelial cells that line the ventricles of the brain and central canal of the spinal cord. They play an important role in the production and circulation of cerebrospinal fluid, or CSF. Microglia are small, elongated cells that have short, irregular processes. Their main function is to respond to infection in the CNS. When activated, they become phagocytic and remove cellular debris associated with damaged nervous tissue. They also secrete immunoregulatory substances, such as cytokines, as part of this response. Oligodendrocytes have large cell bodies with thin, flat processes that wrap repeatedly around portions of axons in the CNS. This wrapping process produces an insulating cover known as the myelin sheath. Each oligodendrocyte can myelinate multiple axons. However, each axon requires multiple oligodendrocytes for complete myelination. Two additional types of glial cells are found only in the PNS: Schwann cells and satellite cells. Schwann cells, also known as neurolemmocytes, myelinate PNS axons. Each Schwann cell forms a myelin sheath around a single axon, unlike oligodendrocytes in the CNS that can myelinate multiple axons. As with oligodendrocytes, multiple Schwann cells are required to myelinate each axon. Satellite cells are small and flat. They provide physical and chemical support for neurons located in ganglia.